looks good. If you have any questions, please feel free to interrupt me or use the chat feature, and I can only see a handful of heads at a time. So if there's anything you want to say, please just say it, and we can make this as much of a discussion as possible. We're also trying a new thing where if you go up to the, oh boy, someone might have to, oh, the annotate option on your screen, you can see a button that says stamp. And if you click on the stamp, then you can stamp the screen with a little question mark or something, that if you don't understand a figure or something, just stamp the screen and I'll stop and we'll talk about it. Or you can ask a question in the chat, or anything you can do to encourage the discussion. Please feel free just to jump in.
I want to point out that it looks a little different from our ends.
Oh, sorry.
Yeah, that's okay. So for those of you who are missing it, if you look at the very top of your screen, you can see you are viewing Kyle's screen. It says view options, and then you can click on that and go down to where it says annotate, and then the little window should pop up so that you can use the stamps.
Sorry, the only time that I use Zoom is for GenSpace. So it's every time there's a learning curve. I shared my screen really effectively this time, so send me applause for that. All right, so how do I advance the slide now that I've...
I think you have to close out the annotation.
Okay, I spoke too soon. My mouse is gone. The mouse is back. And okay, all right, here we go. So if anyone read the, I was scouring the publications that had come out to try and find something that was spooky enough to honor the start of Halloween, or the Halloween season, the spookiest month of the year. And when this paper came out, I thought it was absolutely perfect. And the New York Times article written by Katherine Wu, who is like an up-and-coming science writer, she wrote as the first line of her piece, "When crops have nightmares, they dream of Sclerotinia sclerotiorum." And I thought that was just the perfect line to start with, because this fungus can turn crops like these pea shoots into this, which is obviously totally gross. And not only is it bad for the plants, but because this fungi infects crops that we need to be able to eat and to feed an ever-growing population, this is an enormous concern for farmers and agricultural industry and us in general. So I would amend this first line to say that when farmers have nightmares, they dream of Sclerotinia. And this is because this disease this crop disease has caused routinely up to billions of dollars in damage for the United States agricultural industry. This fungus infects almost 600 different plant species. There are no resistant cultivars to this. So that means that when this Sclerotinia or white mold infects a crop, there's basically nothing they can do. There's no alternate variant of species that farmers can plant that will enable them to continue growing this in their fields. And fungicides, which can be used, have their challenges. They're hard to put on the crops. They don't work very well. And there are also beneficial relationships that plants have with other fungi that the fungicides can wipe out. And so there really are just no good solutions to prevent these types of outbreaks once they happen. So crops are having nightmares about these. Farmers should be having nightmares about these. And I would also add to this that when anyone has a nightmare, they should maybe dream of some particular fungi. This is a review article from Fisher et al. In Nature magazine, and it shows sort of an alarming increasing trend in fungal-related infections and outbreaks that are happening worldwide. So in this A panel right here, you can see that from 1995 to 2010, along the horizontal axes, you can see this really clear trend in the number of plant infecting fungi that are causing these alerts in global monitoring systems. And you can see that these are distributed around the world in this B panel here, and that these are leading to very high numbers of extinction events or events that are causing like significant amounts of damage. So for whatever reason, in the past couple of decades, these plant infecting fungi are increasing. And why in particular this would be a bad thing or something that we would want to stop is hopefully some of the food for thought that can feed our discussions about this paper. So my questions to you guys early on are, why are these trends so scary? And why are fungal infections particularly scary for humans and plants? I mean, we're living in a time right now where obviously we are dealing with a viral pandemic, but why are fungal infections particularly scary for humans and plants? So this, I'll open it up to, I think Juwan was one hand that I saw raised, but anyone feel free to jump in. Oop, I seeing questions on the chat too, but anyway, I'll read those while, Juwan, do you want to jump in?
So the reason, you said, why is it scary, correct?
Mm-hmm.
So, and totally correct me, because like I said, I don't think I know a whole bunch, but I know a lot of the crops we typically have are almost like clones and copies. So typically, if one gets sick, then that means they're all prone to getting sick like the rest of them. And if you can't fix, I guess, one in one area, it doesn't help that you can't fix all of them in any area. And so they're more likely to spread it and pass it, just like how we have a viral disease. It's like, well, they can spread it too, and they'll just keep the trend going. And if they're all bred from the same copy, that means there's nothing different to really protect them. And so then that causes a problem of, well, like, if this one's sick and then gets the rest of the food, then it's all sick. And then at that point, by the time you try to help or figure it out, there's nothing to do to save them. And then you just lose a whole species of whatever.
Yeah, Juwan raises a great point where a lot of the crops that we grow to feed, the billions of people on Earth are all genetically identical. So there's a great example of fungal infections wiping out bananas. So bananas are all clones, they're genetically identical, and several years ago, this species of fungi wiped out every single cultivated banana on Earth, and banana farmers had to switch around, and now the bananas that your grandparents ate are totally different than the bananas that we eat today, because this fungal infection wiped out everything. And there is a new strain of fungi that is threatening to wipe out the current cultivar banana that we use. So it's pretty scary. I think the other thing that is interesting about fungi is that they're eukaryotes just like us. So with bacteria, we can take antibiotics that will affect the bacteria, but not the host as a human, a eukaryote. The same thing with viruses. But with fungi, they're eukaryotes just like us. So even though obviously a mushroom or a yeast cell, pretty different than a human, but they still use almost all of the same machinery. So it's much harder to find antimicrobials that will work against fungi and not kill us too, or plants, which are also eukaryotes. So, and then of course, we need all of these crops to feed us. So there's a lot of issues with decreasing crop productivity and feeding a growing population. Lost my mouse again. Forgive me while I try to find the mouse. Apparently when I go to annotate, it disappears, and I have to find it just by, I'm close. Okay. Whew. Sorry about that. All right. So, you know, I started out talking about a lot of negative things about fungi, but what I would also add to that, just, you know, for to do due diligence, because there are a lot of people at GenSpace who really like fungi, myself included, but in some cases, fungi are not always the enemy, and they can be the hero and come in to save the day, like we'll see in this horror story with a happy ending. So we'll keep in mind the ways that fungi can also be helpful and beneficial to us and to the other organisms that they interact with. Before I move on, I'm going to check the chat. So Madeline asked about something related to carbon in the atmosphere. I'm not sure, without reading through, I'm not sure what that refers to. Madeline?
If that's at all related to the rise across the world of fungi.
Oh, the increase in infections or cases of plant infecting fungi. That's a really good question. I don't think the article that I took that from, Fisher 2012 in Nature, I don't think they deconvolute what might be driving those trends, but you know, when you get right down to it, everything is kind of related to that, and increases in temperature and things like that, land use changes and increasing fertilizer use. So that they could very well be in some way directly or indirectly related. Fungal infection, the potato famine. I actually have no idea what caused the potato famine. It was probably a fungus. I don't know if anyone else knows, feel free to jump in. And yeah, oomycete. Julie, what's an oomycete? Is that a parasite or a fungi?
It's not a fungi, although they were classified as fungi for a really long time. So it's really commonly thought that the potato blight was due to a fungal infection. It's a different, I don't remember how far back, class or order of single-celled species that cause infections largely in plants, a lot like some of the plant and some of the fungi that you're talking about.
But it's a eukaryote, right?
Correct. It is also a eukaryote, yes. No.
Yeah, so they're just, I mean, and even back during the potato famine, they didn't have as much technology to try and to know obviously what was infecting their crops, but we're still today having trouble containing these. And this is, you know, part of the reason why when you go through customs, they make sure you don't have soil on your shoes and stuff, because just walking through these fields can spread these pathogens from one place to another. So it can be bad. Oh, this is my PSA. Not all fungi are bad. Do we have, just for the sake of being the devil's advocate here, does anyone want to jump in with some examples of how fungi are not bad and how they can be beneficial or helpful or commensal to organisms that they interact with? Other than eating them, because fungi are delicious.
There is a comment in the chat that I can read out loud. I'm a parasite from Erie. I'm a parasitologist, so I'm interested in the balance between mutualism and parasitism in biological interactions. I thought this article made a really interesting point in encouraging a more friendly symbiont to establish in an ecological niche as a means of disease control.
Yeah, exactly. So as we'll see, the switch between something that's going to kill you and something that's going to be really beneficial and help you grow can sometimes be this sort of alarmingly thin line that can tip over with just a viral infection. What else do I think of for beneficial fungal interactions? So trees and other plants can have, oh yeah, duh. Beer is a beneficial fungal interaction that enables us to get through these frightening days. But yeah, yeast that rise our bread, the aspergillus that makes sake. I was, of course, thinking like an environmental scientist and thinking of like the fungi that live in tree roots and enable them to get nutrients. In a broader sense, fungi are responsible for breaking down the plant compounds that that no other organism can break down. So were it not for fungi, the earth and all of the life on it would not have evolved as it has, because all of this plant material would have just been built up and no other critters would be able to access those nutrients. And wine too. Yeah, fungi are exactly nature's recyclers. So yeah, they make penicillin, they break down nitrogen compounds. Yeah, they are keeping the elemental cycles of Earth going instead of completely dead-ending after everything has been used. Soil remediation, yeah, so many different positive interactions between fungi and other organisms, but in this case, it starts out not so positive. So we will introduce the cast of characters for this paper first. So the researchers that wrote the paper that we're talking about today, about over a decade ago, they isolated this virus that turns this white rot fungi from this horrible pathogen into a hypovirulent strain, which means that it goes from being super pathogenic to not pathogenic at all. So this group published this in 2010. It's a virus that you can see here in this dark micrograph. Nothing special, looks like a virus, has a really small genome, only 2,166 base pairs, just a handful of genes. But somehow, when this virus infects this fungi, it goes from, when it infects a plant, basically killing it. Here on the right, you can see all of this white fuzzy cloud is the virus killing the plant, or I'm sorry, the fungi killing the plant. When the fungi is inoculated onto the plant that's already been infected with the virus, the plant is totally fine, here on the left. So somehow, when this fungi is infected by this virus, it removes the pathogenicity and it can live on this plant and the plant is totally fine. And these researchers left that here a decade ago. They didn't look into what might be going on molecularly to make this virus tame this fungi. They didn't look at what the potential benefits to the plants are, etc. And that is where this paper works. These open questions about how it works and what potential it has in the field is where this paper picks up. And that is where we are, what we're discussing today. Did I hear someone?
A question.
Yeah,
yep. Question. I was wondering if anybody's studied the connection between the immune system and the fungi activity
in the plants.
Yeah.
Yeah, they didn't in this initial paper, but they do touch very briefly on the plant's immune system in the paper that we read tonight, using only gene expression studies. So it's sort of a proxy for what might be going on in the plant's immune system. And we'll talk about that a bit later too. So that brings us to the paper that we're discussing today. And this paper is sort of broken up into 4 main parts that we'll touch on really briefly and see if anyone has any questions. And then we'll break out into discussion groups to talk about some of the implications and how this paper, the data and results from this paper could be used to help agriculture in the future. So the first part is how the tamed virus, I'm going to keep doing this, I'm going to keep messing up this cast of characters, how the virally tamed fungus can grow inside of plants. So here's, now that we're entering into this part of the talk, if you want to use that annotate feature to put a question mark on any part of these figures, then I'll see it and we can talk more about a particular figure panel or a particular part, or if you just want to put it anywhere to signal that you have a question, then we can talk more about what might be going on in a particular part of the paper. So the first thing these authors did is, oh yeah, we already got a question. I don't know who that is, but if you want to jump on to either the chat or vocally, and I can try and answer. But I might be able to answer this question by what I was going to say anyway, which is that the first thing these authors did is try and figure out what happens when this virally tamed fungus is inoculated into these plants. And what they did is they had a particular strain of fungi that is transformed or genetically modified in a way that makes it essentially glow in the dark. And that's what we see here. All of these different colors, the red and the green, are the fungi glowing in the dark, and then all the parts around it are the roots of a cell. So Amy asks, what is the purpose of using both the WGA staining and RFP labeling? What does each contribute? So if my understanding is correct, the RFP staining allows you to see every single, all of the DNA, I think, of the fungi, and the WGA, or maybe it's the other way around. The RFP is is all of the fungi, and the WGA enables you to see the outline of all of the fungi, or the RFP is the virus. I had the figure legends in here, and then I had to remove them to make space, but we can look at that after to get a better answer to that. But in any case, they used it to see where these fungi were going. And first they looked at just, the baby roots of the plant, and they could see the fungi sort of, minimally invading these plants, growing between the cells. They could see them crossing over into the neighboring cells. And then they could see endophytic growth, which is actually puncturing into the plant cells and really establishing themselves. They compared this to the strain that hadn't been affected by the virus in row D here. And what they found is that the growth was much more extensive, and it was actually killing the plant root cells. Does anyone have any questions on that? Okay. The second part of their paper was aiming to look at picking up where their 2010 paper left off, and looking at how viral infection changes fungal pathogenesis. And they did this by looking at the genes in the virus and the fungi and the plant that were turned on or off. So this figure is looking at fungal gene expression, and across the columns here, you can see the number of hours post-infection. That's what HPI stands for. In DT8, which is the virally infected fungi, and the original fungi that hasn't been infected by a virus on the right. And then each horizontal row is a different gene. And they found a number of intriguing results here in how gene expression in the fungi changed in genes related to plant cell wall degradation. This is the really obvious one, where what they saw was that in virally infected fungi, they saw a major decrease in the ability of these fungi to essentially break down plant cell walls. Obviously, if you're a really pathogenic fungi, you want to essentially eat your host and break down the hard plant cell walls. And these are the genes that are really active when the virus is trying to kill the host, but those turn off in blue after it's infected by a virus. They also saw decreases in effector-like secretory proteins, and these types of proteins are important for suppressing the plant immune response. So I think Randall asked about the plant immune response, and one of the ways that this fungi is so deadly is that it can use these secretory proteins to stop the plant's immune response. So basically have a carte blanche to do whatever they want. They also saw little change in the compound aprosoria, and this is just a fancy way to name the structure in the fungi that's sort of like almost like I guess like a grappling hook that sort of uses this incredible amount of force to puncture through the plant cells. And they saw slightly diminished expression of these genes. And this sort of makes sense because the fungi, even though it's tamed, can still, take hold of the plant, but its ability to do that is slightly depressed. And then finally, they saw variable expression in oxalic acid synthesis. And oxalic acid, they hypothesize, is a function of the fungi that's critical for pathogenicity. And they use this acid to decrease the pH and break down plant cell walls to enable them to take over the plant cells more effectively. In the plant cells, they also looked at gene expression, but what they found is that only about 0.3% of plant genes had differential expression between infection and with one and infection with the other. So not much was different was going on in the 100,000 genes that this plant has. But the important thing that they did notice was that this tamed fungus was actually quite beneficial to the growth of the plants. So when they looked at total plant biomass, they found that plants that were infected with this tamed virus grew more robust, bigger, faster than plants that were not infected with the fungus. So you can see that really clearly in panel D is the tamed fungus infected plants, and panel E is the control. And you can see that these are significantly different values in the aerial parts of plants. Those are the leaves, and the inoculated plants are growing much faster. So this was an indication that this fungus is actually, after being infected by a virus, switching from being this horrific pathogen to actually sparking better growth and becoming an endophyte, or something that is interacting in a beneficial way with the host. And then finally, they also tested if this tamed fungus is beneficial to plants in other ways, in particular, if it decreases the disease severity of other fungal pathogens that could infect this plant. They didn't have a better picture of this in the figures of the paper, but this is a table with a lot of numbers that shows that the disease severity goes down when plants that have already been infected or inoculated with the tamed fungus are infected by a second fungus. It dramatically decreases the severity of the disease that is caused. So I thought of this sort of like taking your probiotics to prevent gut disease or something like that. This tamed fungus is having these beneficial interactions that are protecting this plant from super infections. So finally, I thought we could discuss the last part of this paper in discussion groups about how this dynamic duo can be used like a plant vaccine in the field, and how the researchers tested this, and then a number of other questions or whatever you'd like to talk about branching off of that. But before we break out into discussion groups, does anyone have any questions or comments or things that they didn't understand about the paper, or different lines of inquiry that you might want to talk about as a whole group before we break out?
Kyle, this is Faye.
Hi, Faye.
Hey, I use spent mycelium. There's a guy who grows mushrooms, and I go and pick up his spent blocks, and I use them in my vegetable garden outside to regenerate the soil. And I've noticed that the plants are definitely much more robust, and the aerial parts of the plants, my collards and my greens and oak, all of it is very lush. Mm-hmm. So the question is, I wonder if what they have observed is not necessarily specific to that particular strand of fungi, mm-hmm. but is prevalent with all fungi in terms of mycelium and all of that, the stuff that it does.
Yeah, there are other, that is a great question. So one of the things that I was actually really surprised about from the introduction of this paper is that all of these interactions were actually really specific. So the virus obviously can only infect a particular strain of fungi, and then the fungi are really pretty specialized for interacting with the plant organisms that they either parasitize and kill, or in this case benefit. And I think thinking ahead to some of the discussion questions, maybe that's the main limitation in really applying this to in a biotechnological way or in an agricultural way, is that a lot of these relationships have evolved over evolutionary time to be very, very specific. So you can't just take this particular tamed fungi and then put it on your tree in your garden and hope that your tree grows twice as fast. That being said, with the mycelium blocks that you have, my guess is that those are just a rich source of nutrients. So the fungi in that block have been growing happily and breaking down the compounds as they grow. And then it's like a little block of broken down plant material that is essentially like a fertilizer for your garden. So I bet in that case, the interaction is indirect, but maybe we have a fungal parasitologist here. So maybe I'm totally wrong. So happy to be corrected, but that would be my guess about a worm parasitologist. I'm a microbiologist, so everything bigger than that confuses me. Alrighty, so does anyone else have any questions or comments? Or anything that was confusing about the paper, anything at all?
Someone in the chat asked, how does the tamed fungus affect other organisms, and if so, how?
How does the tamed fungus affect other organisms? So this particular fungus, when it's pathogenic, can infect almost 500 different plants. They didn't test this on any other plants other than rapeseed. So it's unclear whether or not whether it would be able to be scaled to be used on other crops. I don't know enough about plant biology to know how closely, like, are those 450 crops? Does that encompass all of the crops that are important cereal grains or foodstuffs for us? I'm not sure about that. So I don't they haven't tested that yet, but I can imagine that that is a follow-up study that they are trying to do right now. What's cool? I just saw cool in the chat. Alrighty, so if there's no more questions for the whole group, let's break out into discussion groups. How many, am I a host? Am I able to do that?